Hello students, today we are going to study DNA sequencing. DNA sequencing is actually sequencing of the DNA. And what does sequencing mean? Sequencing means you are finding out the order of the nucleotides in a piece of a DNA. Now, from the term order of nucleotide, I mean to say that I need to determine the sequence of amino acids that are forming the strand, which helps in prediction of the protein structure and the function. So, and as you know that any alteration in the DNA sequence can lead to a non-functional protein. So that is why sequencing is an important study. Understanding a particular DNA sequence can, of course, help us in determining the genetic condition. We can differentiate between a gene sequence of a normal cell or a cancer cell. So, DNA sequence is of importance in this technological world, and it has many applications in the field of forensic sciences, environmental studies, and agriculture studies. Now, DNA sequencing is done by two main methods. Sengar, that is first is Sengar dideoxynucleotide chain termination method, which is a commonly used method. And second is the chemical cleavage method by Maxim-Gilbert technique. In this video, we are going to study the Sengar dideoxynucleotide chain termination method. Now, this technique was developed by Fritz Sengar in 1958, and he was actually a chemist who made his first mark in the sequencing proteins, and then he did RNA sequencing also. Now, this method is popularly known as dideoxy sequencing since we use DDNTPs, that is dideoxynucleotide triphosphates, in the as a chain terminators in this method. So, DNA synthesis is done with the help of terminators using dideoxynucleotides as terminators for the elongation in this chain reaction. Apart from the dideoxynucleotides, we require primers, we require the enzyme, that is DNA polymerase, we require template strand, of course, and we require a mixture of all the DNTPs and, of course, a detection system. Now, the synthesized strands are determined for each dideoxynucleotides with the help of electrophoresis, and this is of course an enzymatic method because we are actually synthesizing the strands. This is the pictorial representation of the method. Now, first I would like to discuss the difference between DNTP and DDNTP. Now, as you can see in the two structures, here this is 2 dash and this is 3 dash. Now, in both the cases, the only difference is that at this 3 dash position, in case of DNTPs, that is deoxynucleotide triphosphates, we have the OH group. But in case of DDNTPs, we are lacking with this OH group. That is why they are known as dideoxynucleotide triphosphate. Now, what does this 3 dash OH group makes a difference? The difference is that since this does not have this 3 dash OH group, it will not be able to form the phosphodiester bond with the growing chain. So it means that if DDNTP is incorporated in the chain, it will not be able to form the next phosphodiester bond with the incoming DNTP. And since it will not be able to form the bond, it will act as a chain terminator. So DDNTPs are working as chain terminators in case of Sengar method. So we have coming to the technique, we have the single stranded DNA, we have the radioactively labeled primer, we have the enzyme, we have the DNTPs, and this is how we prepare the 4 reaction mix. So in the first two, we have DDATP, we have DDCTP, we have DDTTP, and in next we have DDGTP. This is the reactions, how the reactions goes in the tubes. Now, for example, if I consider this tube, you can see this strand. Now this strand, we have the primers. Now suppose if this DDGTP gets incorporated with this C, complementary to the C, the chain will get terminate over here. In case G is incorporated here, the chain will get terminate over here. And in case it is incorporated here, we will have the termination here. So these are the three possible products. Now, similarly, we have products from this tube also, and the first tube also. After this, individual tubes have the reaction, we go for gel electrophoresis, and then we go for the autoradiography study. So as you can see in this figure, similarly, as we have discussed for this tube, we have the reactions for this tube also. You can see how the longer fragments are in the upper part of the gel and shorter fragments are moving on the lower side. So this is for DD-ATP, again then DT-CTP, DD-TTP, and for DD-GTP. Of course, this is the sequence of the complementary strand. This was our original strand that we were analyzing. In these reactions, we are actually synthesizing the next strand. So the sequence that we get from here is of the complementary strand. And if we want to get the sequence of the strand that we are analyzing, we have to do a complementary of this sequence to get the actual sequence that we were analyzing. So this is how Sengar sequencing is done. This was all about Sengar sequencing. Thank you.